In this session, we will discuss the Rsamtools package. The Rsamtools package is a library for interfacing to something called the Rsamtools library. This has later been replaced by something called the HGS, or High Throughput Sequencing Library. This is a set of collections for dealing with files in SAM and BAM format. SAM is a text file format for representing aligned reads. BAM is a binary version of SAM, and for practical purposes, because they are faster and more convenient, everybody should exclusively work with BAM files. So that's what I'm going to talk about in the rest of this session. There's not really a big difference with that and SAM files. Let's start off by getting an example before we talk a little bit about the content of the BAM files. So let's load the library and get a path down to an example BAM file. You'll see here that BAM path is just a character vector giving us the path to a BAM file. So the first thing you do when you read in a BAM file is you instantiate a BAM file instance. That is essentially just a wrapper around a pointer. This is a very lightweight class. There's no data in this class. It just contains a pointer down to the BAM file. But you can see there's interesting information such as an index, which is really the magic that makes BAM files fast, and a couple of other settings. At this point, we have not retrieved any data from the BAM file. That's nice in many ways because BAM files can be really big and often we want to think a little bit about before we read them all into memory. Already at this stage, even though it hasn't read anything from the file, there's a couple of high-level things we can query the thing about. For example, we can talk about which sequences or chromosomes were the reads aligned to. The BAM file class here supports seq-info and other friends such as seq levels and seq lengths. So you can see that the reads in this example BAM file has been aligned to two sequences that are roughly 1500 bases long. So BAM files can be really big, and often you want to read the BAM files into chunks. But before we talk about how to read the BAM files into chunks, we will read in the entire BAM files. This is just a small example file, and look a little bit at the output. You read a BAM file using a function called scan BAM. So this returns us something that is a list of element 1. And that seems a little weird. I just asked for reading the entire file. And that's because scan BAM supports reading in different genomic regions. So for example, if you want to read some small or some bins of the different chromosomes, you can read them in here. And this is going to be the outer container on this list here. So the reason why we just have one element in this list is, and we're also going to see it doesn't have a name, is basically that this is the entire files. So let's get rid of the outer, let's subset it to the first element, and now we can see that now we have all the reads. We can see that it's represented as a list. It has 13 different components. Some of them seems to make immediately sense, like strand and perhaps pos, like for position. Let's have a look at the first element of each of these components. If we scroll up here, we can see that Q name is the name for the read. Flag we're going to return to. R name is really the sequence it was aligned to. Strand tells itself. Position is the position of the leftmost part of the alignment. Q width is the length of the read. Here's a mapping quality, something known as a cigar we're going to return to, and then some other things that are interesting. And then here towards the end we get the actual read and the quality values from the read. So the BAM format is quite complicated to talk about because it supports, or what makes it complicated is it supports a rich sense of alignments. So first of all, each read can be aligned to multiple places in the genome, or can contain multiple alignments, or can be associated with multiple alignments. It's also possible for the BAM file to contain unaligned reads. And finally, the reads can be spliced, which is used a lot in RNA sequencing, where in classic DNA sequencing we can think of this as indels, insertions and deletions, where chunks of the read are mapped into different parts of the genome. These things here really is what makes the flexibility of this is what makes the BAM format a little bit unwieldy to deal with if you really want to understand the full horror of the format. In this example here we're showing here, we're going to see some reads that are aligned directly to the genome, and we're going to leave it out to talk about these more complicated representations. If you are going to work with spliced alignments, you really want to look at the genomic alignments package that contains classes for representing spliced reads. Okay, back to BAM files. So let's talk about reading in BAM files in small chunks. You can do this in two ways. You can either read in only parts of the BAM file, in the sense that you read in only certain flags or certain reads that have certain properties, or you can just read them in by saying, I'm going to read the first 10 reads or the first 50 reads. So we're going to start with the last thing first. So we do that by setting a yield size. So we set a yield size on the BAM file, and remember when we print it, we can now see that we have a yield size set here. It's equal to 1. This means that every time we call scan BAM on this BAM file, we're going to get one read back. And for this to work, we have to open the BAM file. So we open the BAM file, and now every time I call scan BAM on the BAM file, I'm going to get a read in. So let's illustrate this. So we are going to call scan BAM on the BAM file, and then we're going to get the first element of this list here, and we're going to look at the sequence. I'm going to call it once, we get a read back, I'm going to call it again, and we get a different read. And this way, I can continue cycle until I get nothing back from the scan BAM command. Let's clean it up a little bit. We're going to close the file. We're going to set the yield size back to NA. Another way of reading in parts of the BAM file is reading in pre-specified regions of the genome or pre-specified components of the file. So let's say we are interested in reading in reads in a specific genomic region that we have represented, or a set of genomic regions that we have represented as a G-ranges object. What we do is we set up a params, a scan BAM param. And scan BAM param is like a class that encodes our query to the BAM file. This is very similar to what we have seen with, for example, BS apply for applying things over genomes and other uses. So here I'm going to start off by setting up a scan BAM param. I give it a width argument, which is what genomic region we're going to read in. And then the what argument, I'm just going to set equal to scan BAM what. This is where we set which flags we're going to read in. So you can see here, this really just returns 13 names, 15 names, and it tells us which pieces of the BAM file we want to read in. This is actually useful because sometimes you're only interested in the position. You don't want to retrieve the actual sequence or the actual base qualities of the read because they're rather big and they take up a lot of space. Okay, so having specified the scan BAM thing, we call scan BAM with the BAM file, and we set param equal to our object we had defined before. And now we see we actually get two components back on the return list, corresponding to the two different ranges we were querying the BAM file in. And then in one of them, let's say the first one, we can see here that all the positions are going to be between 100 and 1500, which looks legit. Note that if we take the head, that they actually have positions that starts for less than 100. That's because the reads are long, and these positions are the leftmost part of the read, so they overlap the position or the G ranges we gave. Now, there's also functionality in Rsamtools for reading in multiple BAM files, because you almost never have only a single file you want to deal with. And this is done through a class called BAM views, which sets up a views-like functionality. So it's really a collection of files. It's a collection of files together with an optional collection of ranges giving us which part of the genome we're interested in. Let's set up an example of a BAM view. And this is going to be a BAM view with only a single file in it. So this is going to appear a little bit weird, because it's going to be very similar to what we had before. But we have no ranges in it. We have one sample, and we can read it. We can read in the data by calling scan BAM on this thing here, and now we get an outer. Now we get again a list, but the first element, the first level of the list is the file name, and then underneath it we had the situation before where we didn't have a name, and then we come back to the actual contents of the BAM file. Now we can set ranges onto a BAM view. So that's called BAM ranges. So now we say BAM ranges, BAM view, and we set that to be equal to the genomic range we had before. And now when we call scan BAM on this BAM views, we're gonna get something that's very similar to what we had before. Let's see here. The outer thing is gonna be the file name, and then when I select the file name, I get what I had before, which is the two different sequences, and then the reads below that. Sometimes you just want a quick summary of what is in the file, and for that there's a command called quick BAM flag summary that very quickly gives you some details of what the file contains in terms of like how often do you have a read that maps to multiple locations, or is a split. So this output also takes a little time to digest, but if you're working or envision yourself working a lot with BAM files, this is stuff that you are going to have to sit down and come to terms with.